All right. I think we're back, and I'm going to go ahead and talk about the fibrous proteins, because they're a topic that's sort of by themselves, and they're not high on the technical detail. They're more high on the recipes and food chemistry. Because of that, it's a topic that we can cover in less depth, and therefore I can give you a lecture and I can count on your book and video lectures, which automatically teach you less than if you're actually here and I'm able to ask you questions and you ask me questions. So we'll do that for fibrous proteins, and then we'll talk about protein folding in class on Monday. Also, if you watch this one out of order, you'll be okay. You won't miss that much, okay? So I do want to start off with this, though. We mentioned I had this in my slides for this next lecture because it was announced last year at exactly this time that Jennifer Doudna won the Nobel Prize. And we had her as the example. Remember that I asked that question of you, how quickly I forget. You know, it's been a long year, right? But at least something good happened in 2020. And Doudna is a member of the Protein Society, which is also something that I maintain my membership in, although I'm not sure in the pandemic if I've let my membership lapse. Okay. I should probably look up those dues and pay the dues. But Protein Society is a real thing. I'm a member, and we have Nobel Prize winners that come from this. Actually, Frances Arnold is another one that won the Nobel Prize. I think she's a member of the Protein Society. I know she's a member of the American Chemical Society. Lots of Nobel Prize winners. And what's really cool about them, I don't see the need. You've probably noticed they're women. And so we have good representation, and I think it's a legacy of Rosalind Franklin and Dorothy Crowfoot Hodgkin, who made the field at least a little more open to women. And I believe the field is reaping those benefits now, because this Nobel Prize winning work is coming from the foundation that they laid. So let me go ahead and talk to you about the globular versus fibrous proteins. Most of what we talk about in this class is globular proteins, but in this video I want to talk about fibrous. Globular proteins are easy to use as chemicals. You just dissolve them up, hen egg white lysozyme. You know, you have the bovine serum albumin. They dissolve, you can get mg per mil of them. And the one thing about them, they're globular. They literally contains the word globs in it, okay? So the globular proteins are, they're folded up, and they must be folded up. Tertiary structure must be a thing that's important, because if you just have secondary structure, then you have a big long string, and that's not globular at all. That's not going to dissolve. For example, let's say you have the same protein 3 different ways. This is human serum albumin, which is 585 residues. And if it was on the beta conformation, you can do that calculation now. You would calculate that it's 2,000 angstroms long by 5 angstroms wide. You're not going to be able to do much chemistry with that. That's going to make a fiber. And there are some proteins that work that way, but they don't form specific chemical shapes that at least we can interrogate easily. You can make, okay, let's make it into an alpha helix, which is a little wider and a little shorter. Still a fiber. If you have an alpha helix and you don't have anything else, you just have this big, long secondary structure that goes off into the distance. But we know from looking at human serum albumin that it is much more like a sphere, much closer to a sphere. So that must mean that these secondary structures are somehow folded up into a compact shape where, instead of being a long fiber, it looks like this. My little friend, this is actually a hemoglobin subunit, but my little friend the globin. We are going to get back to talking about that kind of protein later. But today we're going to talk about the proteins that aren't globular forms, that are more like the fibers, because sometimes you need something to act like a fiber. So globular and fibrous forms, those are the 2 different kinds of forms. So I've got some pictures for you. This is a fibrous protein, magnified greatly, sprouting out of the skin. If you look at it very closely, this is probably clear that some kind of hair is a specific kind of hair. This is actually an eyelash. So eyelashes and other kinds of hair are a specific kind of protein that is fibrous. Its whole purpose is to be a fiber that grows out of your skin. The hair protein is called keratin, and it's one of our 3 main fibrous proteins. Here's another picture of keratin. This is magnified 3,500 times. You still aren't seeing the proteins. So it's a bundle of fibers that has gone together and it might have some kind of coating. But at the heart of each of these hair strands is a fibrous protein. And it has certain characteristics. If you pull on it, it will break at a certain tension. It'll be a certain amount of flexibility. And that will be useful for different purposes. What's really cool about this is keratin has been optimized by evolution for its purpose. And so fibrous proteins usually have structural functions. They're like ropes that tie things together. If we want to study them, chemists can study them and try to figure out how to make materials that might be made out of different atoms but have the same structural benefits of flexibility and or strength. And so we can study these three and look over here when it has the proteins that are studied as biological composites, the proteins that material scientists study. They're the three proteins in gray: collagen, silk, and keratin. Okay. So hair protein at the middle of the hair strand, I think it's the follicle is what makes it. So cross-section of a hair is what it says at the bottom. You've got a bunch of little fibers of alpha keratin that are wound up around each other. Now, the thing about alpha keratin is if you're able to see the actual protein, it's actually an alpha helix. So it's like the case on that first slide where you have just all the sequences, just one big long alpha helix. Now, you do have a two-chain coiled coil of alpha helices that are wound around each other. You see how there's the alpha helix, which is itself a coil. Then you have a coil of the alpha helices, which is a coiled coil, okay? And then those are put into a protofilament, protofibril. Those are biology terms. What we care about as biochemists are alpha helix and coiled coil. So there's the alpha helix, and you know what that looks like. Just remember, coiled coil is a deliberately general term. These are three-stranded coiled coils that are on the front of biochemistry. All that coiled coil means is that you have two levels of coiling, okay? And alpha helix is one of them. All right, so we just showed you eyelashes. This is not eyelashes, but this is actually also keratin. And if you are a biologist, you might be able to recognize this is a feather. So this is keratin, but it's wound up differently. And that's what you can do with fibrous proteins. It matters how they are arranged at the superatomic level, higher than atoms. And keratin is found all over the place. I believe it's fingernails. I believe it's toucan beaks. And if the toucan beak is an optimized structure for cracking open nuts or whatever toucans eat. It's a very strong structure and very light because birds have to be very light to fly. So scientists study those structures. How does it turn protein into a tough, light material that is very strong and that has structural insights at several levels? And so you might be used to styling your hair in some ways. Certain ways of styling hair, including getting a perm, will be doing hair chemistry. And in fact, if you're getting a perm and you're a biochemist and you smell it, it smells like our friends, the reducing agents. Because what some hair chemistry does is it breaks disulfide bonds. So between the alpha helices, you have some cysteines in there, and you have some disulfide bonds. And those are formed a certain way when the hair comes out of follicle. The cool thing is we can be a chemist that will actually break those bonds. We just add a thiol. The bonds break, and then we curl the hair around a curler, and then we oxidize, and the bonds reform. And it stays that way because it's covalently bonded to itself. So what's interesting is hair removers are designed to break all these bonds. And so in a hair remover, you're going to have a thiol. And what you have is you have calcium thioglycolate. So that's what's in hair remover. But you could have predicted the thiol just from first principles. The rest of it is that it's actually a thiol combined with calcium hydroxide. And that's what makes it work really well. The calcium hydroxide does two things. So the basic properties cause the hair to swell, which makes it loosener. And it also deprotonates part of the hair. Think about it. What would it be deprotonating on this slide? You had the thiols. Some of the thiols are reduced. And if you deprotonate them, then you will help to break the hair apart. So you're not just reducing the thiols, but then you take away their protons with a base, and then they can't come back together. The hair itself is easier to break apart at that point. And that's why this chemical is in hair removers. So, of course, hair dyes is just a material. Hair dyes are protein stains, just like your Kumasi blue or your Bradford reagent is actually a protein stain. It turns blue with protein. So if you could actually use it as a hair dye, I guess. I don't think it's safe, so I definitely don't recommend it. But you can have safer molecules that work the same way. They just stick to proteins, and they will stick to your keratin, and they will color it certain colors. What's cool is that some of these can also be pH indicators, which means at low pH they are one color, at high pH they are another. And you can actually look at exactly how the structure changes the color. The one thing I want to point out about this is from blackcurrants, and it actually is, depending on the pH, it's a different color depending on which pH you apply it at. So it's natural, it's safe, it's color changing. It can be different colors, and you probably do cool effects. So, and it's also cheap because it comes from the part that's left over after they make this blackcurrant liquor. Apparently, there's this blackcurrant liquor. I've never heard of it. I don't remember the name, but it's popular in the UK. So all the Brits drink it, but they have a lot of blackcurrant stuff left over, and from that they can extract this hair dye. The only other thing I want to point about these molecules, colored molecules absorb visible light. And notice how molecules that have these collections of bonds, like these pi bond, they're flat, and they just happen to be the energy, the electrons are at the energy where they absorb visible wavelengths. And so you'll tend to have, and if you mess with that structure by protonating or something like that, then you change the wavelength and the color changes. Very cool. All right, so we've had keratin, and this looks like hair again, right? So you might want to think this is keratin. But if you know me, I don't like to be that predictable, and so this is not keratin. Anybody want to think what this is? That would be like keratin, but not. The second most important thing, well, you probably read it in your book. This is collagen. This is actually a lot more painful than hair. This is a ruptured tendon. So collagen shouldn't look like hair. It shouldn't be unwound. When it's ruptured, that means it has some kind of problem. Collagen is used in many places throughout the body. Collagen is stronger than keratin, so it will be used in tendons because the molecular basis of it is stronger. Skin also contains a lot of collagen in it. In fact, if you look at this is a microscopic thing of skin. Skin has many, many layers. And so it's actually a two-dimensional kind of flaky type thing. This actually looks like a croissant to me, but that grosses me out because I don't want to think about a skin croissant. But that really is the closest thing. It has like a million layers, and that's why skin can get flaky and dry and stuff like that. But that's collagen chemistry when you're dealing with that. Now, skin is made to be tough but also to flake. So that's the arrangement of collagen that you get. But in like a tendon or things like this, so this figure shows tendon, skin, and bone collagen. You zoom in on it, and you can see slightly different arrangements of collagen at the 10 to 100 micrometer level. But then when you zoom in, it's all the same once you zoom in beyond that level. When you get to the atomic level, you get to the 10 to 100 nanometer level. And see all the way at the bottom? It's my favorite unit, the 1 angstrom level. And then you have, but you have the collagen structure right here. Collagen is definitely fibrous, and it's made up of what look like alpha helices, but they are not alpha helices. They are triple helices. And if you zoom in and look at them, they are much more stretched out than an alpha helix is. You know an alpha helix is, it goes like this. But these go like this. They go farther than the alpha helix goes for one turn. So they are more stretched out, and like I showed you on the Ramachandran plot, they actually fall into the well next to the beta structure, not to the alpha structure. And if you look very closely, you see that there's, hopefully you can see this, It says glyprohip. That means they are a repeating primary structure of glycine, then proline, then a hydroxyproline, repeated over and over again with only small variations ever. This is what makes collagen so strong, because it has this repeating sequence, glycine, proline, hydroxyproline. And in fact, the best, most universal repeating sequence is glycine, something else, proline. Could be proline, could be something else. So you have this kind of sequence, and this forms a triple helix that is different angles than an alpha helix. It's not going to be hydrogen bonding itself the same way. So that's the question. Why is it so strong? It's not hydrogen bonds because it can't reach its own backbone. When you look down the angle, so you have the side view on the left, and you look down on it on the right. And if you look down the middle, you color all the glycines red and look down the middle. You see all the glycines are in the middle right here. So every third residue must be glycine, because if you look at that, there's no room for any side chains there. You couldn't even have an alanine. It would not be able to fit. Part of what makes collagen so tight is that it wraps around itself to make this tight core that only glycine can fit in, and glycine's flexibility makes a very evenly packed and therefore strong core in the middle of it. So collagen is a very strong thing. The other thing that gives it strength is the proline, sort of restricting it. And collagen is throughout the body in different things. It's in muscles as well. And in fact, different types of meat will have different amounts of collagen. And it turns out that steak meat doesn't have much collagen, and therefore it doesn't have much of the strong protein holding it together. Stew meat does. And that is the whole reason why steaks are more expensive than stew meat. Steaks are more expensive because you only have to cook the outside. You don't really have to cook the inside of the steak, and you can chew it. But stew, on the other hand, if you try to cook stew meat like a steak, you will find that you cannot chew that thing no matter what you do. It's not even edible. What you have to do with stew meat is you have to bring the entire meat cut up to temperature. And what you're doing in that case is you are melting the collagen. You're unfolding it by disrupting those non-covalent interactions enough so that the strands fall apart, and then your teeth can mechanically tear it apart. So collagen is found in different kinds of meat or muscles. It also is a major component of the animal intestine, which means if you had natural hot dogs, congratulations, you used a casing that was made of intestines that was full of collagen. And it was a type of collagen that you could actually chew. Okay, but it's kind of gross. So hot dogs are actually kind of gross if you think about it. I can understand being vegan after reading about hot dogs. So there's another thing that's a form of collagen, gelatin. And so you have probably a lot of my vegan friends know that if you want to avoid animal products, you should avoid those little packets of gelatin. You should avoid Jell-O, because Jell-O is actually protein, and it's made from partial hydrolysis of skin, bones, and connective tissues. Because what that means is that you have all these little bits of collagen that are broken up. They're still strong enough to form the Jell-O matrix, and in fact you can unfold and refold them by adding or removing heat. Okay? So Jell-O is actually purified animal. It's a purified protein solution made from animal. And the other thing you can do is you can do all sorts of chemistry, like if you've ever made Jell-O and you've whisked air into it, then you've let it refold, and it forms just like this gel-like structure where it has this, tangled up things, but you've trapped air bubbles in it. And so then you have the protein Jell-O. There's a reason why Jell-O is a lot like protein crystals. Protein crystals are just a specially ordered form of Jell-O. Jell-O is just where you have all the protein strands. They're all mixed up. You don't care. But a protein crystal will be Jell-O-like because it will be filled with water and made of protein. So this is, by the way, if you want to do this, you can actually do Jell-O inside a Coke bottle, and then you can actually apparently make gummy cola. I haven't done it myself, but the YouTube looks really cool. So the other thing is gelification is also covered in one of these compound interest infographics. I want to zoom in on a couple parts of this because this talks about the exact methods of gelification. So basically you add the protein powder to hot liquid, and then, by the way, if you don't add, if you're adding alcohol to Jello, don't do it before you boil. You've got to boil the water, then add the alcohol, because alcohol is volatile, and you might boil away some of your alcohol. But all that is to say, alcohol is compatible with water, therefore it's compatible with Jello. And the science of what's going on is you have the powder of you have the dispersed collagen particles. You hydrate them, they unfold, and then when you cool them, they refold, but now they're not in particles anymore. The particles have dispersed, and therefore you have a gel that is formed. The other thing that you notice is at the very bottom of that, I said hydroxyproline on one of the slides, because you will find hydroxyproline and even hydroxylysine in collagen. And people have asked, okay, we go through a lot of trouble to have an enzyme that hydroxylates these two things in collagen. Why? Well, it turns out that we have the proof that it's important from just general health, because to hydroxylation is difficult chemistry. You can't do it with just your regular amino acids. You need a vitamin. You need a special structure that you get from citrus fruits, vitamin C, because we evolved in a citrus-rich environment. So we stopped learning how to make vitamin C. We just always ate it. We outsourced it. But then when we start to go on long ocean voyages away from orange trees, then we don't get enough vitamin C. What happens then is that your hydroxylation doesn't happen as much, and your collagen starts to fall apart. The first thing you see with scurvy, well, I want you to think about it a little bit. Scurvy, you start to have your gums bleed. You start to have your skin flake. I think there's something happens with your ears. And then you even start to have cognitive issues. So collagen's important. And and the hydroxylation is important to collagen. So why does it do it? Now, I just have one residue, one paper that explains why this residue is hydroxylated. And you might think it's just a solubility effect, but it's not. It's actually a weird molecular orbital effect, where if you hydroxylate the proline, it will actually flip the ring of it from endo to exo, and that helps flip the backbone from cis to trans. And that the trans backbone is actually important for collagen strength. So that's the best explanation for why you need the OH, but definitely that you need the OH was proved long ago by sailors who didn't get enough of it and got scurvy as a result. By the way, collagen is in skin, so a tumor will disorder the collagen. And there's a scientist, Dr. Bowden, who has detected, she's developing a technique to use light to measure the order in collagen in the skin. And this could detect really small tumors that you can't otherwise see beneath the skin by seeing how they disorder the collagen. If you look at this slide, the bottom picture right here has a tumor, and it's blue because it's disordered. Red means order, and at the top you have no tumor, and red. On the bottom you have disordered collagen because you have a tumor growing in it. And that's actually what she's detecting using light, the order of the collagen. The only other thing is that, of course, chemists have gone in and we've messed with collagen. This form of collagen is really strong. It uses a non-canonical amino acid, which organic chemists can make, but ribosomes cannot. And then you have so it has an azoglycine, but they figured out if I put these azoglycine backbones in there, not only can it fit the right angles, but it also can form multiple hydrogen bonds with the other strand. So they reasoned this out, they tried it, and it worked. Making more bonds makes it stronger. So all this is to say, what collagen strength is increased by hydrogen bonds, but it's so strong, it's got to have some covalent bonds in it. And in fact, it does. And it has stronger covalent bonds than hair does. Hair just has little disulfides, which are easy to break. You just add a little sulfur, and you break the disulfide. But collagen has real, honest-to-goodness lysine hydroxylysine covalent bonds that are made by an enzyme. And these are typical carbon-nitrogen type covalent bonds. These are not disulfides. These are high energy, hard to break. And so you see that the collagen molecules, they are joined together by these. I believe that when you make Jello, you add an enzyme that somehow breaks some of these apart. So what's really cool is gelatin can actually be really useful for other things. There are some scientists that were growing concrete because what we want to do is they have bacteria that will eat CO2 and turn it into calcium carbonate, eat CO2 and turn it into CO3, okay? And they will metabolize that, and so you can not only take away CO2, you can turn it into concrete, and it turns out this concrete is super strong, just has to be grown. They found out that a secret ingredient, just the plain calcium carbonate that they put out, wasn't strong enough. So they added gelatin because it's an easy, cheap, biocompatible thing to do. And they found out that it had sort of a synergistic effect where it became this super strong material because the gelatin made some kind of collagen. They interact and they wrapped around the CaCO3, and it's compatible with the bacteria. They're happy. The concrete's strong. You can make it into any shape. And so that's kind of a cool idea of growing your own concrete out of photosynthetic bacteria and Jello. Here's a little more detail about exactly what they were doing. And so you can pause on this and read it if you want more, but I'm just giving this to you for your information. Here's another one right here. And the best idea is, why would you want to do that? You wouldn't want to do this on Earth, but it would help you to build things in space. Because in space, you can grow bacteria exponentially. You can't necessarily mine calcium carbonate exponentially. So that's collagen. There's one more on our tour of fibrous proteins to talk about, and that is this protein. So it's our Halloween special. This is silk. This is one form of silk. And if you think about silk, definitely fibrous. And if you look at it on a lower level, it turns out to be protein. So you can have a lot of silk. This is not a Halloween decoration. This is a spider infestation of some place. That's a lot of protein, and it's organized. What I can tell you is what you're looking at there is a bunch of beta sheets. So silk, the other ones were, we had an alpha helix, we had a triple helix, and now we have the second kind of secondary structure. We have beta sheets. So silk is smooth. It's not necessarily meant to, it's very elastic, but its strength is not the same kind of strength as a collagen strength. In fact, it's not cross-linked at all. So you don't have any kind of covalent bonds connecting the strands. In fact, what you have is you have beta sandwiches, which have like alanines between them. And so you have a bunch of alanines on top of each other, which means that the silk can move pretty easily. It can move, like the alanines can shift around. And also individual strands can like sort of be pulled out. Stick-slip deformation is what this is called. So this is really cool about silk. And like with the other materials, we have scientists who are studying this. In fact, probably the most scientists are studying silk. And you can all imagine why. They all want to be Spider-Man, okay? It's not 100% true, but it's like 90% true. Because we want to be able to make webs like Spider-Man does. And we want to make a web like a spider does. And really, isn't Spider-Man just an imitation of nature? So we want to imitate nature, and we can imitate it by looking at the molecular structure and making a structure like it. So that's cool. Here's a little infographic just about more about that, and it shows the silk in the fibers just like before. But I want you to focus on this in the middle. The silk has crystalline domains and amorphous domains. The crystalline domains are shown in orange, and they're connected by these spaghetti-like purple amorphous domains. So the silk, this is why the silk is so stretchy, because it has some parts that are like these ordered beta sheets, and some parts that are literally not beta sheets at all. They're just stretchy loops. And it switches back and forth depending on the circumstance. Now what's cool about silk is you actually can eat it. I don't suggest eating a silk, hat or whatever people make silk out of these days. But you can actually make a coating of silk. If you run into a spider web, you're worried about eating the spider, but you shouldn't be worried about eating the web. The web is edible. It's probably not tasty, but it won't kill you. And in fact, you can actually use a silk protein laid out in a particular way, not spun as a web, but laid out as a sheet as food preservation. So here's the sheet about this. And what's cool is that you can see they start with silkworm cocoons, and they dialyze it. They make a solution of the silk protein. And then they dip strawberries in it, and they take them out. And the silk coats the strawberries invisibly with a protein coating, and it's just beta sheets. I mean, don't freak out. It's not really, there's not vitalism going on here. It's just protein, right? Anyways, if I can convince you, maybe you can try one of these strawberries someday. If you have no coating, so you coat them, and you really can't see much difference between the coated and the non-coated. The other cool thing is, depending on how long you anneal them in the water, which means you just leave them in the water at a certain temperature, you will get more and more beta sheets forming. The longer you let it form, the more they will form the beta sheets. This is actually like the egg coating to the paintings that we talked about. Over time, the beta sheet conformation is favored. So you give it more time, it forms more beta sheets. So the other thing is, if you look back and forth, the cool thing about this coating is it will prevent air and other things from oxidizing and messing up your strawberries. So then you have the strawberry that you just leave it out, and if you dip it for longer and have more beta sheets, it will be more protected. And they have the examples of the strawberries that were dipped. The longer they're dipped, the more protected they are, and they stay fresh out on the counter. It works for a banana as well. This is probably the most obvious. After day 9, the banana looks brown, but if you have the silk fibroin coating, it has just a few brown spots. And in fact, they checked its ripeness to see by putting this 200-gram weight on it, and you can see that the brown banana is squished by the 200-gram weight, but the regular banana is still firm and ready to eat. This is probably a little more palatable because you don't eat the banana peel, so you aren't technically eating any of the silk protein. And finally, there is actually even a shoe. I forget which maker. I think this is Adidas. But it's an Adidas shoe made out of a spider silk type material that was woven together by scientists. So the one thing we want to do is we want to use our techniques to try to investigate these. And it turns out that Cryo-EM is a good technique for looking at something flexible like silk. And so they've done some examples, and they've looked at what happens when you stretch silk. In the gland, you make this purple protein, and then you spin it together, and it comes out as this native assembly, which is kind of spherical. You see how you have these purple spheres. But then when you spin it and stretch it, the assemblies aren't spheres anymore. See how they're elongated needles. The beta sheets themselves can deform, and they deform in a very particular way. So they're super strong, but they're also super, they're proteins. They're this common protein materials. They're just amino acids arranged in the right way. So it matters how you physically stretch the sample as you're spinning it to make a strong material. And so this is where they've actually followed this all the way to trying to develop super strong fibers. Now again, we have a gross animal, hagfish. I'm sure they're very nice, but I wouldn't eat a hagfish if it was on a menu. And yet they have really strong protein fibers, and they wanted to see if we could spin these into super strong proteins that would be like Spider-Man's webs. And I want you to see what they did right here. Again, what they have plasmids right here that have the codes for the things, and then you have the dialyzing. I'll be done in just a few minutes, okay? So you have the dialyzed samples, which will then be brought into different parts of different concentrations of urea that are brought down, and depending on how you spin it out, I'll just be done in a few minutes. You can go ahead and come in, but like 2 more minutes, and I'll be done. But feel free to come in and do whatever. So yeah. So what you do is you actually, depending on how you draw them out, you'll have the different arrangements of the structures. And then you will throw in a chemical to cross-link it. Just like what you have for collagen, you can have a chemical cross-linking that will happen. So this is called draw processing. What they do is they just, the draw processing actually manipulates the beta sheets. So you have different arrangements of beta sheets depending on how you draw process it. And so they studied all this, and they tried to see the strength of all this. How do I get a super strong version of this? And what they did is at the end, they got a material that was really strong. If you look at this graph of strength, the farther up you are, the more strong you are. So if you take this hagfish protein and you don't draw process it at all, you just have it be its ordinary sort of mixed up beta structure, you get something that's around down here on the strength. If you draw process it, you actually move it all the way up toward the corner where it says 100% draw processed. And then when you add the cross-link, you bring it up to the red dot. And you see the red dot is almost as strong as the blue dot, which is artificial silk. And so that is what you can do. You can turn a protein into something super strong where you can build all sorts of stuff out of it. And so those are our 3 proteins. And just here's a table that finishes those up. But I just wanted to give you that lecture over the weekend. So thanks for your attention. And I'll see you on Monday.